In this video, we will study the effect of DANA on blood glucose and plasma insulin. So in order to do this experiment, the researchers have did an experiment on the mouse models. So first, to analyze this effect of DANA on blood glucose, they keep the mice on fasting for 24 hours, and next, if when they want to check the effect of DANA on plasma insulin, they keep the mice on fasting for 4 hours. So the left hand side of the results are for the effect of DANA on blood glucose. So these are the results where mice has been kept on fasting for 24 hours. After that, they have injected the glucose for a positive control, and in other case, the DANA at different concentrations are introduced in different mice models. And what they have find out is that with different concentration of mice models, there is with the different concentration of DANA, there is no change in the glucose level. As from the past, uh, as from the previous experiment, they know that DANA will not affect in the case where glucose are not present at a minimum level. And same things they have found for the insulin, where they have checked that in the case where DANA has been introduced or injected at different amount, they do not see any change in the insulin. However, when the glucose is injected, they see the change in the insulin. And also they simultaneously they also check the cytotoxicity of the DANA using LDH, is lactose dehydrogenase, which is usually found in the intercellular parts of the cells. So if the cell membrane get disrupted because of the DANA, then they will be exposed. These intercellular components will be exposed to the will be exposed in the extracellular matrix, where it can be detected by fluorescence. So what they have find out is that up to 10 millimolars, they do not see any change, any cytotoxicity effect. However, above 10 millimolars or 100,000 macromolars, above 10 millimolars, for example, around 100,000 macromolars or 100 millimolars, they see that the cytotoxicity effect of the DANA is predominant in the mice. So the ideal concentration should be either 10 millimolar or less than 10 millimolar. After that, they check the effect of DANA on the plasma insulin levels also. So in that case, what they introduced, what they did is they keep the mice on fasting for 5 hours. After that, they before injecting the DANA, they have injected the insulins to some extent, to minimum to some extent. And what they have find out is that they can see the change of the insulin, effect of the insulin on the blood glucose, that it goes down. However, with the different concentrations of the DANA, they do not see any change in the effect, indicating that DANA does not have any effect on the blood glucose level. And when they check the blood glucose area under the curve, of the results that they did on the panel A, and they get a quantitative analysis that DANA does not have any effect on the glucose level unless insulin uh, is present, unless glucose levels are present beyond glucose or insulins are present beyond a threshold level. Glucose level that we have discussed in our first experiments, that the glucose levels are present around that is, it's not in this slide. However, when the glucose levels are present around 8.3 millimolar, if I am able to recall it correctly, you can check the paper for the correct value. If glucose levels are present beyond 8.3 millimolar, then they can see the effect of DANA on the insulin enhancement. Otherwise, they do not see the effect of DANA on the blood glucose and the plasma insulins that they have checked on the mice models in for both the cases separately. So I hope you understand. This how they validated their effect of that DANA does not have any effect or minimum effect on the blood glucose and plasma insulin uh, in the absence of insulins to a particular level. If you need to understand more about this topic in detail and needs to understand about these experimental details, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.